Good morning, everyone, and thank you for being here today. My name is Dharti. I'm a PhD candidate in Horticulture Science Department, and today I will be presenting my PhD dissertation research. And I'm really excited to share what we have found over the last few years. My PhD dissertation's title is Full Plasma and Ozone Technologies for Irrigation Water Treatment. To understand why this research is important, let me explain some problems that we are trying to solve through this research. So it is very common in greenhouse and hydroponics production to use recirculating irrigation system. And it is great because it has some great benefits. It saves water. It also reduces fertilizer waste. However, it also comes with some problems. The main challenges of recirculating system are pathogen buildup, clogged irrigation lines, nutrient imbalances, and ultimately poor plant health. So what can growers do about it? There are some approaches that are currently used in the industry. UV treatment is one, but it has some limitations. For UV treatment to be effective, the water needs to have very low turbidity so that it can penetrate through the water and then kill the microorganisms. Similarly, chemical disinfectants like chlorine and hydrogen peroxide are widely used in the industry, and they are very effective, but they have some drawbacks as well. These chemical disinfectants can leave chemical residuals in the water, which can over time cause phytotoxicity to the plants, meaning they can damage the plants themselves. Also, it requires purchasing and handling of this consumable chemical. So that brings us to the two technologies that I studied for my PhD, which is cold plasma and ozone. So while ozone has been used in the water treatment for a while, its application in horticulture production is being actively explored, whereas cold plasma is completely new to this context. And they are attractive because they use ambient air and they do not leave any harmful byproducts in the water because they break down into dissolved oxygen, which can actually be beneficial for the plants. And they are environmentally friendly. Let me now explain the science behind these technologies briefly. So ozone is a molecule with 3 oxygen atoms compared to the oxygen, which has 2. And that one extra atom of oxygen makes it really unstable and highly reactive. So it is a very strong oxidizer, which can pull the electrons out of the microbial cell, damage the DNA, it can destroy the proteins, nucleic acid, and other cell organelles, and that's how it kills the microbes. In the water, when ozone is injected, it dissolves in the water, and that's how it stays in the system through the solution, and then it reacts with whatever comes in contact with. Now, cold plasma might be unfamiliar to many people here. Plasma is the fourth state of matter. There's solid, liquid, gas, and there's plasma. So if we apply a very high energy to gas, it creates plasma. So plasma is an ionized gas with free electrons and ions. And there are 2 types of plasma. The first is hot plasma or thermal plasma. And thermal plasma can be really hot, often reaching thousands of degrees of Celsius. And this is the kind of plasma that is used in plasma arc welding or cutting metals. And the second type is non-thermal or cold plasma. This is a plasma that I'm working with. So the overall temperature of the cold plasma remains close to the room temperature. It can be generated at atmospheric pressure with very low energy. Its applications include water treatment, sterilization, air purification. These days, plasma activated water is being actively studied for seed treatments and plant defenses as well. So what happens when we treat the water with these technologies? So both ozone and cold plasma, they create reactive oxygen species. These species are very unstable and they have high oxidizing power. And these species include ozone itself, hydrogen peroxide, hydroxyl radicals, to name a few. So besides adding reactive oxygen species, these technologies can also increase the dissolved oxygen in the water, which is beneficial for root health, especially in hydroponic system. Since these ozone and cold plasma technologies are very strong oxidizers, they can damage the cell membrane, cell wall, and other cell organelles and kill the microbes in the water. But besides microbes, they can also react with other organic and inorganic compounds that are present in the water. For example, pesticide residues or even fertilizer killers. Major advantage of these two technologies is that when they react in the water, after they have done their job, they will break down into oxygen. So they do not leave any harmful residuals in water. Compare that to other sanitizers like chlorine, which can cause phytotoxicity at higher concentration. So this picture here is taken from Dr. Rosario Dowley's experiment, and we can see clearly at 0.5 and 1 ppm, there's visible damage to the roots as well as shoots. Now the picture on the right are of the hemp plants that I grew in deep water culture system. These plants were grown in full plasma treated water at 500 MB ORP and 800 MB ORP. Before I move into next slide, I want to explain what ORP is, because I'll be using that term a lot throughout this presentation. ORP stands for oxidation reduction potential, and it is a proxy that we use to measure the oxidizing strength of these technologies. So a higher ORP, such as 800 MB, means it has a higher oxidizing power, and a lower ORP means lower oxidizing power. And as we can see on the picture here, even at a high ORP of 800 millivolts, it did not cause any phytotoxic effects to the plants. Now, this is where things get a little more complex. So, cold plasma and ozone, their mode of action is non-selective, which means if they can damage the bad guys in the water, like biofilm and pathogens, they might also affect the good guys in the water. For example, beneficial microorganisms that we add to help plant, or even micronutrients in the nutrient source. So, understanding and managing that trade-off was the central theme of my PhD dissertation. So, with that context, the overall objective of my dissertation was to evaluate cold plasma and ozone technologies for irrigation water treatment in recirculating horticulture system, with emphasis on micronutrient solubility, microbial quantification, pathogen inactivation, and compatibility with beneficial microorganisms. Each of these points became a chapter of my thesis, and I'll walk you through each one now. So the first chapter addressed the micronutrient solubility. With this chapter, our objective was to evaluate the effects of cold plasma and ozone on oxidation of iron and manganese in the nutrient solution. Our second objective was to compare the oxidative demand of various iron forms in nutrient solution when treated by cold plasma or ozone. So we wanted to see which of the iron chelate was more resistant or susceptible to being oxidized. We used a randomized complete block design for this experiment. We had two treatment factors. Factor 1 was four different kinds of iron chelates, including iron EDTA, iron DTPA, iron EDDHA, and iron HB. And our second factor was water treatment, including cold plasma or ozone. On the left-hand side is the picture of the cold plasma that we're using in our lab, and on the right-hand side is the ozone treatment system. And I'll explain briefly how this system works. So this cold plasma unit is called iron solution, and it's from the company Ingersoll Rand. In the reservoir is where we have our nutrient solution or water. And then this white box, so inside this white box is the plasma chamber. So this plasma system, it uses ambient air as feeding gas. So air passes through an oxygen concentrator, which is inside this plasma chamber. And that oxygen concentrator, it gets rid of all the nitrogen that's present in the ambient air. So now we have an oxygen concentrated air, which then passes through the plasma chamber, where a very high voltage electrical current is applied to the gas, and that creates the plasma. And then the water, in this case nutrient solution, is pumped to the plasma chamber, where the nutrient solution comes in contact with the plasma, and the plasma injects all the reactive oxygen species to the water. And that's how we get the solution that has high dissolved oxygen and also high ORP. Ozone on the right hand side, it works similarly. There are a few components that are different. So the black box here is the oxygen concentrator. This oxygen concentrator, it provides oxygen gas to the ozone generator. So this ozone generator converts the oxygen molecule into ozone molecule. So now we have ozone gas that is then injected to the solution through a Venturi injector. And then now the nutrient solution with the ozone gas then passes to this big contact vessel. This contact vessel is pressurized. The purpose of this contact vessel is to better mix the ozone gas into the water. So now we have the ozonated water that comes out of the contact vessel. So the nutrient solution was recirculated through either cold plasma or ozone until our target ORP of 700 millivolt was achieved. And during the recirculation, we were collecting our nutrient sample from two different points, one from the reservoir, and then we're also collecting the samples coming immediately out of the injector or the reactor. And the sample pairs were collected every 5 minutes. And the collected samples were then analyzed for soluble iron and manganese using a colorimeter, and they were also analyzed for total chelated iron using a spectrophotometer. So this graph shows the time it required for each chelate treatment combination to reach our target ORP of 700 millivolts. On the x-axis, we have different iron chelate types, and on y-axis, we have time in minutes. The first thing to notice here is FeEDTA, which required the shortest amount of time to reach 700 millivolts. And FeHBED in cold plasma treatment required the longest time to reach our target ORP. And this is important because the time it required for the solution to reach target ORP reflects the oxidative demand of that solution. So in the solution with a high oxidative demand like EDDHA or HBED, it means that it is consuming the reactive oxygen species, which is making it difficult for these treatment technologies to raise the ORP. And that also means the more time it takes for the technologies to treat this solution, which we can see on the graphs here. So this slide shows the decrease in the percent chelated iron from the initial concentration. On the x-axis, we have ORP, whereas on the y-axis, we have the chelated iron in percentage. The first graph represents iron EDTA. It was the iron chelate that took the fastest to get to the 700 ORP, and it was also the chelate that was oxidized the least. And if we compare that to graph C and graph D, which is HBED and EDDHA, we can see there's a drastic loss in the chelated iron in the solution by the time it reached our target ORP. And that means when we are treating our nutrient solution with the ozone or cold plasma, using iron EDTA might be better because it is more compatible with these technologies compared to HBED or EDDHA. No matter which iron chelate we use, we will have to resupplement some amount of this after they are treated by these technologies. So the key takeaway from this chapter is that both ozone and cold plasma are strong oxidizer, and they can oxidize the micronutrients present in the nutrient solution. Switching to iron EDTA might be a better idea if you're using these technologies, just so that you'll have to replenish it less compared to other iron chelates. All right, moving on to chapter 2 now, which address the microbial quantification techniques in water treated with ozone or cold plasma. So traditionally, the standard for quantifying microbes in the water is culturing them on agar media, which requires collecting the sample, plating the sample, and then waiting for a few days for them to grow, and then count the colonies. And it works well in lab conditions, but in a greenhouse or a field system, it might not be very practical because it is time consuming. It takes a few days to get the results. It also requires some level of lab training. It requires a dedicated space, and it might be very challenging to maintain a aseptic condition in the greenhouse, which is why growers need faster, simpler alternative techniques that they can use in the field. And that leads us to two alternative techniques. The first one is Pythium, which is a dehydrated culture media, and it provides some level of selectivity for the target organisms. For example, you can buy Pythium that will quantify aerobic bacteria, or that could quantify just yeast and mold. So basically, it is a ready-made nutrient media, and it usually has a plastic cover sleeve, and it also has some indicator that makes counting easier. ATP test is even faster. ATP stands for adenosine triphosphate, and it is an energy molecule found in every living being. So if there's a living organism in the water, it will have some ATP. And the commercial luminometer, which is what we use to test ATP, they're very fast. We usually get the results within a minute. So ATP might be a reliable on-site tool for rapid microbial quantification, and that is what we were trying to test with this experiment. So the overall objective of this chapter was to compare ATP and Pythium methods for microbial water quality assessment, and also evaluate microbial control by cold plasma and ozone across different ORP levels. For this experiment, we collected lake water from Lake Alice, and we recirculated the lake water through ozone or cold plasma, and we were collecting the water sample from two different points, one from the reservoir and one immediately after the injector at different ORP levels. And the collected samples were then analyzed for aerobic bacteria and yeast and mold counts using Pythium, and also for ATP measure using Hygiena insular luminum. So for the Pythium measurement, we use two kinds of Pythium, aerobic Pythium and yeast and mold Pythium. First, we collect our water sample in a sterile bottle. For aerobic bacteria count, we diluted it to 1 to 100 dilution using sterile water, whereas for yeast and mold, we plated it undiluted. And then the 1 milliliter of the sample was pipetted onto the Pythium plate, and the spreader plate is used to spread it evenly across the Pythium. And then we incubate the plates in the dark for 3 days. And after 3 days, we will see the colonies forming on it, which you can see on the picture 5 there. Each red dot represents one colony forming unit of aerobic bacteria. For ATP, we use a Hygiena luminometer and its accompanying free and total ATP swab. Total ATP measures all the ATP that are present in the water, including the ATP inside the living cells or any ATP that might be free floating outside the living organisms, whereas free ATP measures the ATP that is free floating outside the living organism. And in this case, what we were interested was microbial ATP. And to get the microbial ATP, you subtract the value of free ATP from total ATP. So we first collect the water sample, and then we make 2 small allocates of the water sample to test total ATP and free ATP. And then we dip our ATP swab in the solution for about 2 to 3 seconds. And then on the top of the swab, there is a bulb. Inside that bulb is the reagent. We break the bulb and then squeeze the reagent so that the reagent flows through the tube and comes in contact with our water sample. And then after that, the swab is inserted into the luminometer, and then within 15 seconds, you get your ATP values in terms of relative likelihood unit. So this slides the decrease in aerobic bacteria and yeast and mold counts with increasing in ORP with ozone and cold plasma. On the left-hand side, we have Pythium aerobic counts expressed in terms of survival ratio. And we can see with the increasing ORP level, there is a decrease in aerobic bacteria counts. And at around 500 millivolt, we see complete inactivation of aerobic bacteria. Whereas yeast and mold were slightly more resistant, and it required and about 600 millivolts to get the same level of control. Both ozone and cold plasma had similar level of control at same ORP level. So this scatter plot shows the microbial ATP as a function ORP. And we saw the similar trend with microbial ATP as well, where with the increase in ORP, there was a consistent decrease in microbial ATP. And at around 600 millivolt, there was a complete decrease in microbial ATP. One of the key questions of this chapter was whether ATP correlates well with our Pythium value or not. And this table shows the correlation between those two. We got a moderate but a positive significant correlation, meaning they are not identical, but they can be complementary with each other. And the key takeaway from this chapter is that ATP could be used as a reliable on-site measurement tool. So if growers want to get their microbial assessment in real time, ATP could be a really good tool for them. Moving on to chapter 3 now, which was evaluating the effects of water treatment technologies on pathogen inactivation. Specifically, we looked at Pythium afanidermatum, which is a destructive pathogen in both hydroponics and greenhouse production. Within this chapter, we had 3 different experiments. The first experiment was short-term exposure assay. Second experiment was reservoir inoculation and single pass exposure. And third experiment was reservoir inoculation in a recirculated system. Within the short-term exposure assay, we had 3 different water treatments, including ozone, cold plasma, and oxygenation. For ozone and cold plasma, we had different treatment levels, including the ORP from untreated to 900, whereas for oxygenation, the treatment levels were 5, 10, and 18 mg per liter of dissolved oxygen. So we treated our DI with this water treatment system, and then we collected the DI water at different ORP level or dissolved oxygen level. And then we added our zoospore suspension to the bottle or to the treated water to get the final concentration of 2500 zoospores per milliliter. And then we let the solution sit at room temperature for either 10 minutes or 2 hours. And after 10 minutes and 2 hours of contact time, we plated 100 microliter of the sample on selective PAR2 media. And then we incubated the place in incubator at 25 degrees Celsius. And after about 24 hours, we counted the number of colonies that were forming on those plates. So this is looking at the change in recoverable CFUs of Pythium with increasing ORP level with ozone and cold plasma. The top graph is for 10-minute contact time, and the bottom graph shows the 2-hour contact time period. We have ORP levels on x-axis, and on y-axis we have CFUs of Pythium expressed in log scale. So across both contact time, we can see for ozone at 700 millivolts, there was a significant reduction of Pythium CFUs, whereas for cold plasma, it required slightly higher ORP of 800 millivolts to get a significant reduction. However, at 900 millivolts, both treatment resulted in complete inactivation of Pythium afanidermatum zoospores. But if we look at the oxygenation data, we did not see any effects on the inactivation of Pythium. There we can see there are the different dissolved oxygen level on the x-axis. On the y-axis, we have recoverable CFUs, and we can see there was no significant difference among those 3 dissolved oxygen level on CFUs of Pythium. And this is a very important message because this tells us whatever inactivation we got from cold plasma or ozone wasn't a result of elevated dissolved oxygen. It was the reactive oxygen species like ozone that was actually doing the work. That being said, maintaining a dissolved oxygen level is really good for health. It improves the root health. However, we cannot expect elevated dissolved oxygen to work as a sanitizer and control the pathogens that are present in the water. Now, this is experiment 2 within our chapter 3. In this setup, we had DI in the reservoir where we inoculated the DI with the Pythium zoospore suspension. So the solution with the Pythium zoospore was then passed through the ozone or cold plasma or oxygenation system once, and then we collected the sample immediately after it came out of the system. The question here was how much reduction can we get through a single pass through this system? And this table summarizes the result from our experiment 2. If you look at the percent reduction there, with ozone, we got 100% reduction even through a single pass through the system. Whereas for cold plasma, we got a significant but incomplete reduction of about 68%. And as expected, we did not get any significant reduction with oxygenation. The difference in the percent reduction with cold plasma and ozone could reflect the differences in the units that we are using. The ozone system that we are using, it injects a higher level of dissolved ozone compared to cold plasma. And it could also be the differences in the oxidants they produce, the flow rate, and etc. So in this case, with the condition tested, we can say ozone was more effective compared to cold plasma, and with oxygenation, there was no control of Pythium. Now in the experiment 3, we had the same reservoir with the DI that was inoculated with Pythium zoospores, and then we treated it through either cold plasma or ozone. We did not use oxygenation because we did not see any effects of oxygenation in previous 2 trials. So the Pythium inoculated solution was recirculated through ozone or cold plasma, and we were collecting samples out of the reservoir at different ORP levels. And we were also recording the cumulative ozone level in the reservoir at different ORP levels. In this graph shows the reduction in Pythium at different ORP levels in a recirculation system. And the vertical dashed line represents the ORP where we first detected the complete inactivation of Pythium, which was approximately 400 millivolts. So now if we compare that to our first experiment, which was the short-term exposure, it required 900 millivolts to get the complete inactivation. But in a recirculation system, a lower ORP of 400 millivolts was enough to get the same level of control. So the picture on the bottom is visually showing the same results. We can see at 400 millivolts, there's no Pythium growing on the plate. Even though it looks like a lower ORP was enough to kill the Pythium in this system. However, it's really important to know that in recirculation system, even before it reaches 400 millivolts, the solution has already done a couple of tank turnovers. It has already passed through the ozone or cold plasma a couple of times before it reaches 400 millivolts, which we can see on the graph B there. So by the time the cold plasma reached 400 millivolts, it had already done 4 tank turnovers. Whereas for ozone, by the time it reached 400 millivolts, it had done 2 tank turnovers. So that means it has already been exposed to the ozone or other reactive oxygen species a couple of times, and that reactive oxygen species exposure has already killed the zoospores. And since we were also recording the dissolved ozone in the reservoir, we can see by the time the cold plasma reached 400 millivolts, it had already accumulated 2.1 milligrams per liter of ozone, whereas for ozone system, it was already 1.1 milligram per liter of ozone. So, and those dissolved ozone levels were probably enough to kill the Pythium zoospores in the conditions that we tested. So, the takeaway from this chapter is that treatment efficacy will be different based on how you treat your solution and based on how your system is configured. In a one-time exposure, a higher ORP might need to get the same level of control, whereas in a recirculation system where the solution is being continuously treated, a lower ORP might be effective. So it is really important to track other things like dissolved ozone level and also look at the system configuration besides just measuring the ORP to get the complete idea of the treatment efficacy. Now we move on to the fourth chapter, which was evaluating the compatibility between beneficial microorganisms and water treatments. Within the chapter, we have two different kinds of experiments. Experiment one is tank mixing. Within the tank mixing experiment, we have two different trials. And then we have experiment two, which was container substrate. So beneficial microorganisms like trichoderma and bacillus are being widely used in horticulture, and they are used to improve plant health. They are used as biofungicides to protect plants against diseases. And like I mentioned before, the mode of action of these technologies are non-selective. So our question with this chapter was, if these water treatment systems can kill the pathogens that are present in the water, can they also affect the beneficial microorganisms that are present in the water? So the objective of this chapter was to evaluate the effects of commonly used water treatment system across a range of concentration that was relevant for pathogen control on commercially available formulations of bacillus and trichoderma. So this is the first trial within the tank mixing experiment. In this trial, we had two water treatment systems, ozone and oxygenation. And for the beneficial microbial treatment, we had two commercially available beneficial microbial products. First product was root seal, which contains trichoderma species. And the second product was seeds, which contains Bacillus subtilis. Both these products are from Bioworks, and they were applied according to the manufacturer's recommended high rate. So the reservoir had a dye which was inoculated with the beneficial microbial product, and then the solution was recirculated through either ozone or oxygenation, and we were tracking ORP labels or DO labels in the reservoir, and we were collecting our water samples at different target ORP or target DO labels. And then the samples that we collected were then plated on selective media. We had a different media for quantifying bacillus, which you can see on the left-hand side, and on the right-hand side is the selective media that we used for quantifying trichoderma. So this is the result showing the change in CFUs of Bacillus subtilis on the top and trichoderma on the bottom, with increasing in ORP. And we can see a very clear trend here, where with increasing ORP, we see a decrease in CFUs of these organisms. And the table on the side is the regression analysis. And we can see for both microorganisms, we got a negative slope, which was significant, which means a negative slope means with increase in ORP, there was a decrease in CFUs. So the important message from this experiment is that if growers are using these technologies to treat their water, and if the beneficial microbes are getting exposed to this water treatment directly in solution, it will reduce CFUs or populations of this microorganism. In contrast, oxygenation treatment did not reduce any CFUs of these organisms, which was expected because oxygenation does not add any reactive oxygen species, so it is not doing any sanitizing activity. That means oxygenation is compatible with beneficial microorganisms, including Bacillus and Trichoderma. And we can use oxygenation alongside our biological control program without any interference from oxygenation. We wanted to broaden our water sanitizer technology that we use. So besides ozone, we also added sodium hypochlorite, hydrogen peroxide peracetic acid, a quaternary ammonium chloride product, and we wanted to test the compatibility of these four treatments with the same beneficial microorganism. Trichoderma and Bacillus. So within each water treatment, we had different concentration or different ORP labels, and these concentrations were selected based on the concentration that are used to control pathogens, especially Pythium. And these concentrations reflect the concentration that growers would actually use in their production system. So in this experiment, we had a 500 milliliter flask, which was our experimental unit. So each 500 milliliter flask had DI water in it, and then we made our sanitizer solution in the flask to get the target concentration. And then we added our beneficial microbial product to the flask, and then we let the solution sit at room temperature for 1 hour. And after 1 hour contact time, we first measured the sanitizer concentration, and then we plated the sample on selected media for microbial quantification of Bacillus and Trichoderma. So these 4 graphs show the effects of 4 water treatments on Bacillus CFU. The first graph is for sodium hypochlorite. On x-axis, we have different concentration of sodium hypochlorite. On y-axis, we have CFUs of Bacillus expressed in log scale. And we can see at 2 mg per liter and 4 mg per liter, there was significant reduction of Bacillus CFU compared to untreated 0 mg per liter and 1 mg per liter sodium hypochlorite. Similar trends were seen with hydrogen peroxide PAA treatment. We can see at higher concentration, there was significant reduction of Bacillus CFUs. The highest concentration actually resulted in 100% reduction. The graph C is for ozone. We have different ORP levels on x-axis, and we have CFUs on y-axis. And we can see that at higher ORP levels of 600 to 800 millivolts, there was some significant reduction. However, the degree of reduction wasn't as great if we compare that to sodium hypochlorite or hydrogen peroxide. And the graph D is for the effects of QAC. Similar to ozone, we saw some significant reduction with QAC, especially at 1, 7, and 17 mg per liter. However, if we compare that to sodium hypochlorite and H2O2, the degree wasn't as. We saw similar trends in the effects water sanitizer on Trichoderma CFU. Again, sodium hypochlorite resulted in significant reduction at 2 and 4 mg per liter compared to 0 and 1 mg per liter. With H2O2, we saw significant reduction at the highest concentration. With ozone as well, we saw the similar trend where at higher ORP of 700 and 800 millivolts, there was significant reduction compared to control. And with QAC, again, we saw a significant reduction, especially at 1 and 17 mg per liter. So the overall takeaway from this experiment is that if we expose water sanitizer to beneficial microorganisms in solution, they will cause some significant reduction in the populations of these beneficial microorganisms. However, sensitivity might be different depending on the water sanitizer concentration or the microbial organisms that we are using. Now moving on to where we tested the compatibility of water sanitizer with beneficial microorganisms in a container greenhouse production. For this experiment, we transplanted 4-week-old cuttings of poinsettia in 6-inch pot, and then we applied the beneficial microorganism product as substrate drench on the day of transplanting, and again 7 days after that. We had 3 beneficial microbial treatment for this. We had root cell, which contains Trichoderma, seeds, which contains Bacillus, and we also had a no inoculation control for which we were using dye water. So we started applying our water sanitizers 3 weeks after transplanting because we wanted to give the beneficial microorganisms enough time to get colonized and established in the roots. And for this experiment, we had 5 water treatments, including sodium hypochlorite, hydrogen excuse me, I made a mistake here. It should be S2O2. The hydrogen peroxide PAA. We had QAC, ozone, and DI control. And the active ingredient for sodium hypochlorite was 2 mg per liter of chlorine. For gerotol, it was 114 ppm of hydrogen peroxide. For QAC, the target active ingredient was 7 ppm of QAC. For ozone, it was 2 mg per liter of ozone, or around 700 ORP. And then we also had a DI control. And the water treatments were applied through irrigation for a period of 2 weeks. So we were applying the water treatment 3 times a week. So the experiment was carried out for 2 weeks after we started applying the water sanitizer. And at the end of the experiment, we took 2 kinds of data: microbial quantification data and plant growth measurements. For quantifying the microbes in the roots, we took 5 grams of roots and substrate mix, and then we put the root and substrate mix in the saline solution, and the solution with the root and substrate were then put on the shaker for about 3 hours, so that the microbes in the roots would get dislodged in the solution. And then we took 100 microliter of that solution, and then we plated them on selective media for bacillus and trichoderma. And you can see the colonies of bacillus are growing on the selective media here. We also collected some plant growth measurements, including plant height, chlorophyll index using SPAD, root and shoot fresh weight, shoot dry weight. Before I move on to the microbial data, there's some important information about these sanitizers when they pass through the substrate and root zone. So through one of the irrigation, we also collected some extra leachate so that we could measure the sanitizer that were present in the leachate. And this table summarizes the result from that irrigation. So we have the concentration of the sanitizer in applied solution, and then we also have the concentration of sanitizer in the leachate. And we can see for each of these water treatments, there is a drastic reduction of sanitizer as they pass through the root and substrate zone. So that tells us when the sanitizer is applied through the substrate and when it passes through the substrate and root zone, the substrate acts as oxidant sink, especially the organic matter. It consumes a lot of sanitizer, the reactive oxygen species, and that is why you can see such a drastic reduction in sanitizer concentration as it comes out of the root zone or the substrate. And that has a very important implication, as we can see on these results here. So this is the graph showing the reduction in colony forming units of bacillus and trichoderma in the root and substrate system. And we can see there is no significant reduction with any of the water treatments on either of these organisms. So if we compare that to the tank mixing experiment, where we saw a significant reduction in both of these microorganisms with all the water treatments, whereas in this system, it did not cause any significant reduction. The results that I showed you on the previous slide actually explains this, because even though we applied sanitizers with high concentration as it passed through the system and reached to the beneficial microorganism, the active ingredient was so low that it did not affect the viability of these microorganisms. Now, this is the graph showing the plant height and root fresh weight with different water treatments. So we can see ozone and hydrogen peroxide, the greatest plant height compared to sodium hypochlorite and QAC. Similar trends were seen for the root fresh weight, where ozone resulted in the highest root fresh weight if we compare that to sodium hypochlorite and QAC. And this could be because even at low concentration of sodium hypochlorite and QAC, it could have caused some phytotoxic effects to the plant, whereas ozone, it usually dissociates into oxygen, which is beneficial for the plants. So this result could be twofold. It could be results of the beneficial effects of the ozone and hydrogen peroxide, and it could also be the phytotoxic effects from sodium hypochlorite and QAC on the plants. Now, this is looking at the interaction effects of water sanitizer and microbial treatment on soot trace weight. On the x-axis, we have water treatments within each microbial treatment, and on the y-axis, we have soot trace weight in grams. The main thing to notice here is a lot of the treatments are sharing the same letters. A lot of the treatments have ABC, which means most of the treatments did not result in any significant differences in terms of soot trace weight. The one bar or one treatment combination that stands out is ozone with no inoculation treatment. That resulted in the greatest soot trace weight, whereas the sodium hypochlorite in Bacillus subtilis treatment resulted in the lowest soot trace weight. So it can be said that the water treatment effects depends on the type of water treatment as well as the beneficial microbial treatment. But the overall message from the plant growth data is that ozone and hydrogen peroxide are definitely more compatible with the beneficial microbial treatments compared to other water treatments that we used. Now, let me bring everything together from all the chapters. So the first point or the first takeaway is that both whole plasma and ozone improve microbial water quality. They control the aerobic bacteria and yeast and mold counts present in the surface water, as well as inactivated Pythium zoospores. The results for Pythium zoospores, it varied depending on the system. Under recirculating system, a lower ORP was enough, whereas in short-term exposure, a higher ORP was needed to get the same level of control, which means system configuration is as important as our ORP or ozone or our concentration of the water treatments. Treatment efficacy depended on ORP, contact time, and system configuration. So it is really important to see how those systems, how these treatments are set up before we set up our target ORP or target ozone. Both these treatments had non-selective mode of action, so they affected iron chelase, including iron chelase and manganese, and they also affected beneficial microorganisms during direct exposure, whereas under the container substrate production, the substrate provided some buffer, and since the substrate consumed a lot of the sanitizer, the beneficial microorganisms and water sanitizers were more compatible. So the last and the most important point is that the effective integration of these systems require a system-based approach. It's really important to monitor ORP, monitor the sanitizer residuals, also monitor the micronutrients level and supplement micronutrients when needed, and then timing biological application to avoid direct sanitizer exposure is important. With that, I want to thank all the people who've made this work possible. Very sincere thanks to Paul, our research advisor, for his support and guidance and encouragement. Thank you for the opportunity, knowledge, and confidence you have given me throughout this past couple of years. A special thanks to my committee members, Dr. Nunes, Dr. Swati, and Dr. Perez for their guidance and also for their feedback and encouragement. Very special thanks to all the people in FISR team. A special thanks to my undergraduate helper, Heather and Michael, and also Pili for all the help you have provided through all these experiments. It would not have been possible without you guys. Also, Ingersoll Rand for providing us the whole plasma technology, the funding. It has been a wonderful experience working with them. All the industry partners in Floriculture Research Alliance, Dr. Rosa Rodales, who provided me training during the third chapter with the Pythium, and then also our greenhouse crew, Cody and Brian, and then everyone in the horticulture department. Thank you very much, and also thank you for being here today.